So I guess the best way to start is probably just to see if there's any generalized questions. Is there anything you guys had issues with as far as trying to get through all the terminology and bone markings? I will, of course, you can type in the chat or feel free. You should have access to turn on your speaker. Everybody's okay with that?
Oh,
So,
I have a question. I'm sorry.
go ahead.
I have a question.
Hit me.
Okay, awesome. Okay, so I finished the written quizzes obviously like last night. But I didn't know. Okay, so like on the syllabus and on the calendar that you gave us, it only says that the Quizlet on terminology was due by midnight. But then I saw on Blackboard that the terminology and the bone markings were due, which was like underneath, I guess, like the folders or something like that. And so I was just a little bit confused. I
Yeah, I was going to combine those into one Quizlet, but it made it close to like 90 some 98 terms, so I kept it as two separate ones. I must have forgot to mention it to you guys. I mentioned it to the group yesterday that they were two separate ones to make them a little less overwhelming, but they were still both for the same due date yesterday. But
got
I mean, fine. Yes, there was a misunderstanding, so it's fine. You guys having a couple more days to get that one done,
okay. I
and
finished
I
it.
can send out an email to everybody again to remind you that just please do that one. Yeah, I know I was supposed to combine them together when I made the syllabus, but I decided after looking at that it might be more manageable if you weren't staring at 90 terms, and not that 70 and 20 were a whole lot better, but
Okay, thank you. And then you
so
also
let me make a
mentioned
note
that
of that. Go ahead.
on the Blackboard quizzes and exams, you said that the spelling matters.
Correct.
What about like added S's? You know, because I've noticed like during the quizzes, like there's like articulation versus like articulations, like with like an S at the end. Like, will that count off if I like accidentally put an S there?
Sorry, Shlem, my computer is losing its signal, so I missed that last piece of your. Shalom, I missed that last piece of what you said. Sorry, my computer signal is being wonky. I have terrible cable. So, okay,
Gotcha.
you were saying about Blackboard quizzes. That's where I got cut out.
Okay, you said that on Blackboard quizzes and exams, the spelling matters. And there's some words where like on the Quizlet there was like an S at the end, like articulation versus like articulations. And I know it's like the
Yeah, so the biggest thing is try to watch for the plurals. And usually when I do the Quizlets, or not the Quizlet, I mean the Blackboard quizzes, I will put in singular and plural as both correct answers. And on things like the terminology, there's the possessive form and then just the regular noun, which is usually the S versus like the AR or something ending. I
Mhm.
will put both of those in there. I tried to
Okay.
get you guys to focus on the bold terms on the terminology, but Quizlet is not currently doing it that way. I didn't have time to go back and check and make sure all that is fine tuned. So either possessive or the regular forms I will give you credit for. So parentheses or bold, just don't give me ear head for the
Okay.
terminology. So I try
Gotcha.
to watch for some of that. And all the quizzes and exams are regraded by me.
Mhm.
So the Blackboard it's a computer program. It's black or white. It's either right or wrong. So
Mhm.
I will go back and I will look for any questions that I feel I can give credit to. So
Okay.
please be comforted by the fact that if if there's something super minor or some glitchy thing with the computer program, I will go back and regrade everything. It probably takes me a week or so to get them regraded, but I will go back and regrade the quizzes and,
Okay.
of course, the exams on Black.
And lastly, I know that you gave us like a lot of label figures and like files, just like blanks on them that we can fill out. Is there like an answer sheet for them so that I could like, is it for practice or are we gonna eventually turn them in? Like,
No, it's just for practice, and the picture should relate directly towards the OpenStax
gotcha.
pictures.
Okay.
So no, I do not have any keys for those. Generally, it's just pull open some textbook and use those pictures, which should be the identical kind of things
Okay,
on them.
thank you.
That's just for practice. If you don't like, if you would prefer to do paper versions versus doing Quizlet or something electronic.
Gotcha. Okay, thank you.
Everybody else okay? So this is considered lab 2, so we're moving on to tissues, tissue types, and the structures within the tissues, which I'll pull up this lecture here in just a minute. So Quizlet is already posted for that one. And then I'm working on, and I'll send you guys out an email as soon as the Blackboard quizzes are created and up and ready to go. I lost her yesterday, so I'm kind of scrambling. I'm behind on some things. And then let's see. Of course, I should have pulled up the calendar or printed it out in front of me. I was trying to think what else you have due. So I think that's it for this week is Quizlet, and then your Blackboard quizzes, which are due on Sunday, which I will have posted where you have at least a couple of days to do those. So it'll easily be up by Friday, but hopefully sooner than that. So you guys can take those when you feel comfortable to do so. Is there anything else that I missed? I think that's pretty much it. We don't throw in visible body until next week. So if you're having trouble with visible body, just let me know and we can pull that up and practice with that on these sessions. Because we do the visible body next Monday with the skin integumentary. So I'll pull that up again next week when we talk about skin. And then, and also remind you about tagging, which again, you don't have to do for another week or more. And then just don't forget, I know it's fast, but the exam, mini exam, because it's less points than our big exams, starts next week. So you can't get behind on this stuff. So keep going, keep going. That's the problem about summer. I tried so hard to warn you guys to make sure you're prepared for the fact that we're double pace. So what you usually have a week to do, you've got 3 or 4 days to do before you come back and get material. So, and yes, I also try to warn you that unfortunately lecture and lab are not going to coincide right now. I do not know what you guys are doing. Generally, Dr. Grillo will start with the chemistry, the basic stuff, and I'm trying to figure out what other topics he'll start out with. So he won't really get into tissue probably until next week sometime. So we're pretty much always going to be a week ahead of lecture, and there's just no way around that. So unfortunately, it doesn't give you a whole lot of nice in sync overlap, but hopefully he'll start on tissues before the quiz on it. Or not quiz, sorry, the mini exam on it next week. But it may not work that way, I'm afraid. So generally he'll do more physiology and we'll do more of the anatomy. So that's unfortunately never perfectly in sync either. So summer's a weird setting. There's no way to slow us down to keep us up with lecture. It's just too fast paced. So we just have to do the best we can. Okay, so let's go through and talk about tissues. So I'm sure in your years of doing stuff, and especially even in high school, a lot of high school teachers got through some of this material, that you see over here on the right-hand side, that you have your levels of organization, that you know we start from the little bitty and work our ways up to the bigger. So atoms, molecules, cells, and then tissues are made up of different cell types. And of course, those tissues together make up organs. And then we'll go through the organ system. And of course, the organisms that we're focusing on are humans here. So there are 4 main types of tissues. They're classified as epithelium, connective, muscle, and nervous tissue. So tissues are cells that are related together as function. And the main basic functions of each of these 4 types of tissues are epithelial tissues, which is the skin is one of the major ones you get about when you think about epithelial tissues. The outside of your skin is that layers of epithelial tissue, so it's covering stuff. The connective tissue is holding things together or supporting it. And of course, we know muscles are what give us movement, and the nervous system are our control system. It tells everything what to do. And when we get into different organs, generally they'll have multiple of these tissues working together to function as a unit. Connective tissue is specialized because it has a extracellular matrix, which you'll see here on some of the microscope slides here in a second. So looking at first epithelial tissues, the nice thing about the epithelial tissues is the names of the tissues are based on how many layers they have and the shape of the cells. So simple epithelium means one layer of cells, stratified means more than one. So their names tell you something about them. So that should make it easier to name them than some of the other tissues where you just have to kind of memorize their name. And then the second word that goes with it is they're based on their shape. So the squamous epithelial are kind of look like scales, they're kind of squashed, they're tapered at the ends and bigger in the middle, which is this first picture here. And then cuboidal cells are cube-esque. I say esque just because they're not perfectly square, because human cells always have rounded edges. Plant cells are actually really, really square, but humans are rounded, but still it will be pretty much same size on all sides. They will be similar on the sides, versus if you look at columnar here, one side is going to be tall and the other side is going to be shorter. So columnar are columns. I mean, it's tall, skinny. It's the beginning of its name, so it shouldn't be too hard. And I think most of us from math know what a cube looks like. So that's the nice thing about epithelial. It actually gives you what it looks like in its name. So as long as you can translate those terms, you've got the name of the tissue. So this is just, you won't have to memorize all this. In lecture, you'll go through and have to learn all the locations to where these tissues are. But this picture is just kind of nicely trying to summarize some of the different locations. And the key thing to think about with epithelial tissue is the function goes along with the shape of the cell. So if your cells are really thin and you only have one layer, like the simple squamous cells, that's going to be something that's not super protective. Its job is going to be allow things to come and go, because it's a thin layer. It's not going to block stuff. It's going to allow things to transport. So you can kind of think about that in your lungs. It allows that exchange of gas between your lungs and the blood. Then as things get thicker, they generally are trying to block things out, but still have some control about what goes in and out. So things like the columnar, which are bigger, and stratified columnar, which we're not actually going to make you know because there aren't a whole lot of tissues to have those, much more control, blocks things out. So there's not as much movement between the inside and the outside of those cells. When you have lots of layers, the nice thing about lots of layers is that it allows you to lose some of the layers or have the layers get damaged and still the tissues be protected. So if you think about our skin, which is stratified squamous, the outside layer is dead and is sloughing off. So it can take the roughness of us touching it and the sun shining on it, and it can fall off and our body is still protected. So the amount of layers tell you something about what their function is, even though we won't focus on that in here, just because sometimes it really helps to understand why things are created when you're learning stuff, so it's not just all rote memorization. So simple squamous is kind of hard to section through. So generally you'll just see kind of a top view of it, like this one on the right-hand side here, because it's very difficult to section through these tiny thin layers here. They succeeded in doing it on this one, but generally a lot of times you'll just have to see it from the top, because when it's so many cells like that, it's really hard to get any machine to cut through them. So it can be kind of confusing because you're looking at this top view. You may think that maybe it's cuboidal, but just be careful because it's got way more than 4 sides. These are more hexagonal usually in shape. But this is simple squamous epithelium. So within each of the tissues, sometimes we'll have specialized structures, but in the epithelial tissues, there really isn't a whole lot of specialized structures. But besides the fact that you have to know their name, I am asking you one basic location. We're not going to go into ton of details, just one simple location. The lecture will get into way more in the physiology of this kind of stuff. So the key place where the simple squamous epithelium is in the lungs, so it allows that exchange of gases. Then simple cuboidal epithelium. So the key thing here is when you're trying to look for the cells, is look for the brightly colored nucleus. Not every person who stains is going to use the same exact stain. So you can't go by color. Color is going to fail you because there are hundreds of different stains that people will use to stain these tissues. So you got to go for shape. You got to go for different appearance things, not color, because sometimes it'll be purple, sometimes it'll be black, sometimes it'll be pink, sometimes it'll be red, because there are a ton of different stains. But you will always have the nucleus be stained generally a darker color. So look for the nucleus to help you kind of define where the cells are. See, unfortunately, I told you cuboidal is round-esque, but it's not tall and skinny. There's only one layer here. This is cavity, so you can see that there's just one layer of cells here. And square-esque. Columnar cells are way taller, and the other tissue types, muscles, connective, nervous, are significantly different in shape than these. So there will not be anything that's super close to this. I'll show you what I mean when we get to the other tissues. But one layer, because this is the end of the layer here. This is the inside of a tubule. Most likely this is kidney, because that's the main thing that I want you to note for. But you can kind of see that it's about even on each side. So it's not going to be perfectly a box. Like here's a prettier one you can see, more boxy shaped. But look for the nucleus, because that'll tell you where one cell starts and the next one stops. So its location is the kidney tubules or kidneys. Tubules is very specific to a part inside the kidney, because there are other, of course, tissue types within a kidney. So unfortunately, when I'm running this slideshow, I cannot see Blackboard, Collaborate, Ultra. So if you guys are posting questions, please do them via your microphone. Otherwise, I won't be able to get back to the chat box until after I've gone through all the slides. I can't bounce between the two. Once I play the PowerPoint, it blocks out Blackboard Collaborate. So just to warn you guys, if you have questions, just yell them out. Otherwise, I'll have to go to the chat box and pull this back up again after I'm done with all the slides. So look at here. This is the columnar. Again, simple, meaning one layer. But if you look at them, they're much taller and skinnier. This is actually not the nucleus. These little globby things in here are called goblet cells. So the nucleus are these little purple guys at the bottom here. But if you look at this, it's long and skinny. Long and skinny, yeah, because this is white in color. So if it's white, it will never be the nucleus. The nucleus will always stay darker purple in color. So here you can see a really nice nucleus. And so you can see that this is tall and skinny. The columnar cells, for whatever reason, the nuclei are all at the bottom of the cell, not dead center. So tall and skinny, tall and skinny. This is important in the intestine, and the function of these little pieces here, goblet cells, are to actually secrete mucus. So you've got food going through this cavity, and it could be really harsh and abrasive on the inside of the tissue if you didn't have something to protect it. So that's what mucus does. It helps moisturize and protect. So we are asking you to know one little specialized structure in this epithelial tissue. The rest of them, the other ones, you won't have to know any specialized structures. In epithelial, you will for connective, muscle, and nervous, but for epithelial, the only specialized structure that we care about, because most of them don't have any extra structure, they're just one layer cell or two layer cells, is the goblet cells, and that's very specialized, secreting mucus inside the intestine wall. They kind of look like little eggs to me. So for a lot of these microscope slides, I highly recommend that you draw them out and figure out what key features you can focus in on. Again, remembering color is not a good choice. Don't do color. So now we're getting into the ones that have multiple layers. We really only ask you to do one stratified. So this is stratified squamous. So if you focus in on the nucleus again, you will see they're long, kind of tapered, squashed looking cells here. Scale-like is what squamous means. And you can see there's a ton of them all stacked on top of each other. So there could be, stratified could be 2 or 3 layers. It can be 15 or 20. So this is very characteristic of our skin. When we do the integumentary skin model, which is next Monday, which will be, I guess, our next lab, you'll see what those layers look like, and we'll discuss very briefly what the function of those are, because each section has a little different of a function. Of course, the outside one is dead, and it's so that we can be rough on it and not harm ourselves. And then the lower layers are the developing, growing cells that are moving up as the cells slough off. So this is why you need so many layers, so that the outside ones are damaged. It's basically, you got warriors here, if you will. You've got your frontline troops here, and then the supporting troops. As these get injured, they're going to move up and take their place. So stratified squamous and skin. And there is, I forgot to mention that every week there will be the lab handout sheet that has all these words on it on Blackboard. So you can print it out and you'll have this as a checklist. This PowerPoint slide is posted on Blackboard. We're in Lab 2 activities now. So now we're moving on to connective tissue. So connective tissue, the key to connective tissue, if you think about the word connective, is something is holding these cells together. And generally it's a matrix. The matrix can be fluid, it can be solid, or it can be kind of in between, kind of a gelatinous. And most of them, it's kind of that in between. There's only one that has a fluid matrix, which is blood. So blood cells, of course, are fluid matrix, is that plasma. So you've got, so it's inside blood vessels. So you have white blood cells, red blood cells, and then that fluid matrix, in this case, is the plasma, that liquid that these cells are floating around in. So white blood cells are pretty distinctive because they have nice dark colored nuclei in here. And then our red blood cells, which you'll learn when you get into A&P 2 and learn more in detail about this, but I'm sure you guys, again, probably learned basics of this somewhere along your lifetime, general biology or high school. Mature red blood cells do not have a nucleus. Inside our bone marrow, they do. They develop and can reproduce like regular cells with nuclei, but once they're mature, the nucleus is gone so that they have the ability to absorb more oxygen in the hemoglobin. So they've got weird little dips in the middle, why you can kind of see that some spots are a little darker than others, because that's where it's kind of synced in. That makes red blood cells kind of unique in the fact that they're cells, but they don't have those dark nuclei, because once they're mature, they can't reproduce anymore. So this one, I'm hoping, and the part of the reason about popping up these is you could also use this kind of as a quizzing system when you download the PowerPoint if you wish to use it. Of course, Quizlet will be usable also, but the rings on this should be a giveaway. So bone is very distinctive in that it looks like tree rings. So I'm sure bone is inside the skeleton. It makes it inside the skeleton. All our bones, which are part of our skeleton, have bone connective tissue in them. But these rings are very distinctive to bone. No other tissue looks anything like that. It's the only one that looks like tree rings. And this is also the only one that has a solid matrix to it. So the bone cells, which are called osteocytes, they're trapped. Once the calcified material builds around them, they're trapped inside there. They can't move around. So they have to send signals to each other through little connecting pieces, which are called cannulae. You'll get that in lecture. And here we're not going to ask that much detail. And here we want you to know that the bone cells are called osteocytes. So all these little black spaces here house those osteocytes. Then there's a hole in the muscles, the nervous tissue, the lymphatic tissue to get in. So there's no way to get anything in or out without drilling a hole through this bony material or creating a hole. Then there are generally somewhere between 6 or 8 rings on each of these little circular units, and the rings are called lamella. Okay, so that's just the basic stuff. You'll learn way more detail in lecture when you get to bones in a couple weeks from now. It's really kind of a cool but complex system that works here, but the cells are basically in their own little jail cell. They're trapped, and so they have to communicate through other means because they can't move around. So blood is the only one, the liquid matrix, bone is the only the solid matrix, and everybody else is more of a gelatinous jelly-like matrix. So we'll move on to those. So vivid colors makes it nice to look at, but unfortunately the colors are not always the exact same colors. But the key here is in hyaline cartilage that you have these little things that I always think kind of look like eyeballs, because this is nucleus. It's just, it has dark ring, but it's not as dark in the center. And it has this kind of jelly bean, eyeball-y, somebody said they thought it looks like footprints, whatever you want to describe it as. These are the cells that are in there. So let's first talk about where it's located so I can start pointing out the structures in here. So the trachea, so that's another thing you'll learn about in A&P 2. Unfortunately, this stuff is systems you won't get until A&P 2. But the trachea is that windpipe, so I'm sure you guys heard that term before. So it goes down your front of your throat here, you can feel it. It's got little rings, and those little rings are made out of cartilage. So it allows it to be flexible and strong at the same time. So the structures you have to know inside the hyaline cartilage are pretty much always, we're going to ask you to know the cell type, the main cell type within there. So chondrocytes, chondro is just Latin for cartilage. So chondrocytes are those things that look like eyeballs or footprints or whatever you want to describe them as. And then the matrix in this case is that kind of jelly-like material. It doesn't have anything in it. The reason why I'm pointing that out is I'll show you here with a couple of the other connective tissues. There's no other specialized structures within that matrix that help hold it together. It's just that kind of purple jelly. Not true for some of the other cartilages, though. That's how they make them distinct. So pick out the weird little eyeballs, which are chondrocytes. You know it's a cartilage. And then look at the matrix to figure out which of the other cartilages it might be. If it's not have any other specialized stuff in there, it's hyaline. Now this one here is elastic cartilage. So again, still has the weird little cells that kind of look like eyeballs. But if you look here, what do you see that's different about the matrix? So the matrix is not just that purple hue. It's got this weird kind of fuzzy to it. So first of all, elastic cartilage, if you think about its name, elastic, where in your body do you have very flexible pieces of your body? So the example we're asking you to know is external ear. So very stretchy, flexible. If you think about the word elastic, you can understand why it got its name. So now the specialized structures are, it's a cartilage, so it's still going to have chondrocytes, or those eyeballs. Whoops, wrong direction. It's the matrix is the space between it, but there's something specialized in that space that the other cartilage did not have. Those are the elastic fibers. Those little hairs connect everything together and make it a little stronger in the fact that it can bend and give more than the hyaline cartilage. would be able to do. So you can really jiggle and wiggle your ears so you can kind of pull on them and stretch them, because these fibers are allowing for that movement. They're holding it together enough that it can withstand that bending and stretching. So it looks similar to cartilage, except for it looks fuzzy. It's got those kind of hair-like structures in the matrix. Now the last cartilage is a very specialized cartilage, which is called the fibrocartilage. So it is located in your inner vertebral discs, so those cushions in between your vertebrae to help take that force as you're bouncing up and down. And we'll learn way more about the vertebrae when we get to that section here in a couple of weeks. So still has the chondrocytes, they're just not as close together. There's only a few of them, they're still eyeball-y looking. There's a few here and another kind of buried in here, and there's another one kind of hiding up here. But they're much further apart than either the hyaline cartilage or the elastic cartilage. So their matrix, you don't see a whole lot of gaps left in it because they have these nicely, very organized collagen fibers in there. So they're dense and protective and squishy, if you will, because they have these layers on top of layers of these collagen fibers. So different from the elastic in the fact that the elastic, the fibers go in all directions and kind of look hairy. The collagen fibers are very nice, laid on top of each other in a very orderly structure. They're in layers side to side. They don't go in all directions. And there's less chondrocytes. And it does not have that fluid matrix like the hyaline cartilage had. So hopefully you can see that even though they have similarities, there's still distinctive differences between each of the cartilages. So the last connective tissue here is the adipose, or it's, I think there might be one more. Of course, I don't have my list in front of me. The adipose connective tissue is fat. So fat, oh yeah, no, there's a couple more. So fat is housed inside these little ball-like structures. So this is all fat. Adipose is just a scientific term for fat. So if you look here carefully, the nuclei are shoved into kind of the corners here because the fat pushes all the cells to the side because it fills in the space. So the cells are actually like shoved off to the side because the middle is all just fatty tissue. And it's considered loose connective tissue because there are gaps in it. It's not thick and dense. There's spaces. The spaces in this case are filled with fat. So adipose loose connective tissue is everywhere. Unfortunately, for many of us, we would prefer that there were some locations it would not deposit itself. But it has a function. It's energy storage, which for us, when we have access to food pretty much all the time, that's not a huge concern anymore. But it was when we were back in the day foraging in the wild. It also protects our organs, acts as a cushion, so it's pretty much everywhere. Insulation underneath the skin, it helps keep in heat so we don't get cold as fast. So there are nuclei here, there, and everywhere. They're just shoved to the corners. And then the fat droplet is what's on the inside of those cells, which that space inside the cell is why it's considered loose connective tissue. Versus you'll see in a second that we have dense connective tissue also. So that's a loose connective tissue. This one is also a loose connective tissue. It's easier to see because the spaces are a little more obvious. The other ones, the spaces are the fat droplets. Here you can see that it's not filled in. There are gaps in between the fibers and the cells in here. So this is areolar loose connective tissue. So it's kind of a basement membrane that makes up underneath the epithelial tissue. Helps hold the stratified squamous tissue of things like the skin to things like our muscle that are even deeper. But you can see it's loose because there's lots of gaps. And there are a couple of these that are similar, but the key here again is looking at the things they have in common and then the things that are different. So loose connective tissue has fibrocytes, so that's their cells, and it gets the name fibro because it's going to be making fibers, and the fibers are the pink and darker purple lines on here. So thin versus thick, because the colors are never perfect. In every slide there may be slightly different color, but the thickness is your key here. It's the thinner fibers are those elastic fibers. So some give to it, helps hold things together. And then the thicker, fatter cells are the collagen fibers. So you have collagen and elastic fibers in there. If you think about collagen, that's one that they talk about a lot when they talk about our skin, because that's underneath our epithelial tissues, underneath the basement ones, which is what helps keep our skin kind of taut. And when we get older, it starts to get stretchier and there's less collagen, so it gets looser and our skin starts to wrinkle. These are those collagen fibers when they talk about trying to get collagen injections and collagen creams and that kind of stuff. It's just trying to plump up and rebuild these collagen fibers, which help hold our skin in that nice young appearance. So that's areolar loose connective. So that's generally our skin kind of holding everything together. Now this one's very specialized. So this is called reticular loose connective. So it still has the same fibrocytes, but first let's tell you about where it's located. So lymphatic organs. So lymph tissue has a lot of function with filtering and immune. So this is only in a couple of those organs. So not super common, but still has a good function. It still has fibrocytes, but if you can see, the fibrocytes are way more abundant than it was in that areolar loose connective tissue. So it kind of looks to me like grapes on a grapevine, because they're just so many little bundles and then just the darker colored strands that run through it. So these are specialized fibers. They're called reticular fibers. If you look at them compared to that areolar loose connective tissue, this is much more like a tree or a net. They're all kind of attached and going in all directions, versus the collagen and the elastic fibers are long, skinny pieces, very disorganized, and they're individual strands. They don't interweave or hook or branch with each other. They're just long, flat, one-directional lines, versus this, you can see it's moving and it's kind of branching here and branching there. So that's very unique to just the reticular loose connective, but still has those kind of purple dots, which are the fibrocytes. Now it's a little more organized because now we're getting into tissue that's considered dense. It doesn't have those big gaps in it. Yes, when you slice through it, you're going to get a little bit of gaps in here, but generally that's more of an artifact of trying to cut through the tissue. But you can see here the spaces are very limited, and you've got these long kind of flowing pieces of tissue fibers coming down here. So really compact, dense, hence why it's dense regular. The term regular means that it's organized up and down. It doesn't go in all directions. There is a dense tissue that kind of goes here, there, and everywhere, which is irregular. So this, because it's nice and organized and dense, makes it strong. It can be in things like the tendons and ligaments, which of course we need to be strong. It attaches and holds our bones and things together, and our muscles. So you need to have these be dense and strong. So you can see there's cells in here. They're not as common again, but they're in there, and they're what helps produce these fibers. And to make them strong, we don't want our tendons and ligaments to stretch, so we use collagen fibers, which are the strong ones, not the elastic fibers, which are the ones that are allow for bending and flexing, because our tendons and our ligaments need to hold our joints together. If you've ever popped a joint out of socket, you would prefer if it didn't stretch. So this is dense, regular connective tissue. So holding together things like our joints and holding our muscles to our bones. So that is the connective tissues. So now we're moving on to our third classification, which is our muscle tissues. I probably should have put a category header for these, which I should maybe go back. But so this is type 3 of the tissues, which is our muscular tissues. And there are 3 types of those. There's the skeletal muscle tissue, which I'm sure you've heard before. Skeletal muscle tissue is our muscles that cause the movement. They're the ones attached to the bones and allow us to move. You'll learn way more detail about this, but I'm sure you know because they're skeletal muscle, they're voluntary, which makes them special compared to the other 2 types. So we actually have to tell our muscles to move. They don't just do it spontaneously, like the heart that contracts. They are striated. In lecture, you'll get into a lot of detail about the striation because the striation is very important in how the muscle contraction occurs. But as far as identifying it on a microscope slide, it makes it easier to define it from the other 2 types of muscle. So very nice, beautiful stripes here. Very organized. Cardiac muscle also has stripes, but they have branches. They'll actually curve and go different directions. The skeletal muscle is just nice, organized. They're layered on top of each other, all in one direction, because those muscles have to work together. If they were going in all different directions, you wouldn't be able to create your arm bending. It would try to bend left and right, and some of it wants to bend straight. So you need to have them working as a unit, so they have to be going in the same direction to cause that motion. So there are nuclei in all cells, so that's pretty much a giveaway. We just remind you of that when we do our tissues. And then this long, skinny piece is called the muscle cell. There are not as nice defining edges to that, so just realize that this nucleus is in the middle of one long piece here, which is all one muscle cell. And I already mentioned the striations. So all muscles, of course, are going to have their own cell type. In this case, it's a muscle cell, so it's not too hard. The name just tells you it's in the muscle. And the striations is just that key point to help remember that from some of the other muscle tissues that don't have striations. So they all have something unique about them. Striations not unique because cardiac also has it, but they're the only muscle tissue that will go in a very orderly direction. And I'll show you that here in a second. And generally there won't be huge gaps between here. Again, when you cut through muscles, it's difficult to make them stay put. So this is cardiac muscle. So I'm sure you can get by the name that cardiac muscle is in the heart. These gaps are not artificial. Those are true. You see here, this one's a nice example that yes, it still has the striations, but you can see that this piece here, so the ends are actually these specialized tissue here. I'll tell you in a second. But you can see that you got a piece that goes this way and a piece that goes that way. It's still all one muscle cell, just it's branched. Skeletal muscle will not be branched. The branching is very unique to cardiac, which helps with the electrical signal, so your heart will actually contract. So those specialized tissues at the end of the muscle cells here are called intercalated discs. Unfortunately, you won't get this kind of information until you get into A&P2, but just realize those intercalated discs are unique to cardiac muscle and have to do with the electrical signal that tells the muscle to contract. And the physiology you'll get to when you get to A&P2. But that is a specialized structure that will help you identify that the branching and those intercalated discs should help you not to accidentally call it skeletal muscle. There's still muscle cells, which in this case is easier to see because they're divided by these little darker lines. and they are striated just like skeletal was. And that striation is important in muscle contraction, which Dr. Grilly will talk about if you have lecture here in a couple weeks. The last one is smooth muscle. So not striated. It still contracts, it just contracts in a different way, so it doesn't need those striations. Smooth muscle are in hollow organs. So hollow organ is just a generic way of saying things that have a space in between them. So if you think about your organs, what ones are hollow organs? As like your esophagus, where your food goes down through the middle, is a hollow organ. Your stomach is a hollow organ. Your intestine is a hollow organ. So most of it is inside the organs that you think of as your digestive system, but it is also other places besides just the digestive tract. But that's the majority of where it is in the digestive tract, because it will squeeze to push your food along. It will squeeze to help churn and move your food in the stomach, and of course then move it through the intestine until you excrete it out of the body. So the muscle cells here are squamous kind of shaped. They're tapered. They're fatter in the middle and skinnier at the ends. So if you were going to accidentally confuse this with anything, I can see how possibly you would confuse it with the stratified squamous. But these cells are way longer than the stratified squamous cells are. Stratified squamous cells are they still tapered, but they're shorter. They're only like yay big, versus if you look at this one goes here and all the way over here. It's much larger. So hopefully you can tell the difference. Also, the texture of them is a little different just because the cells inside them are a little different. But this is smooth muscle, so that's the muscle cell. It's long and skinny with the ends being pointed. Of course, all the darker colored things are nuclei. You've got those in every cell that is able to reproduce. As we mentioned, there's an exception to that rule. The red blood cells lose their nuclei, but every cell starts with a nuclei. Some of them will just lose them. Like platelets. Platelets don't have nuclei when they're mature also, but they're still cells because they start out with a nuclei. So now we're moving on to our last classification, which is nervous tissue. So nervous tissue, brain, nerves, I think we know where the nervous system runs through our body. The nervous system here, the key thing to look at is these neurons. Compared to the other cells in the other body, these are kind of star-shaped because they have all these little legs, if you will, that go off different directions. Of course, those are what's sending the signal between the cells and moving the signal down your body. Then they have these little purple dots that are in the matrix, the space between those. Those are the support cells. So they help bring in nutrients and they help destroy things that are not supposed to be there. Whoops, okay. I couldn't remember if I made you learn any of the other smaller stuff, but no, that would be something we would do once we get the nervous system. So key here is we're just trying to pick up some of the key cell types, the neuron, and then the neuroglia, which are all our little purple dots. The rest of these other strands in here we'll talk about when we get to nervous system. Should be the fact that neither tissue has anything that's kind of star, or I don't know how else to describe it. It's got pieces branching off on all directions. And the neuroglia just itself are specialized because they're just little dots. They have no other structure around them. Like the epithelial tissue are boxes, so there's a nucleus in the middle and then some matrix around the cell. This is just a dot. That is the cell. It is just that dot. There is nothing else that's surrounding it. It has no other walls around it. So that is connected tissue. These pictures are on Blackboard, so I'm going to stop sharing this for now so I can get back to Blackboard Collaborates. This slideshow is on Blackboard, so you can use it to review if you want to practice. Of course, there's also Quizlet to go along with it. I will be using these pictures for the Blackboard quiz and for the exam. Now, I did not use these exact pictures on Quizlet. I probably need to go back and fix it. I partially did that because I didn't want you guys to memorize the picture. I wanted you to memorize the appearance and the key features, because you really need to be able to learn this stuff no matter what the stain is, no matter where the person took the section. So if you get too reliant on just memorizing the pictures, you're not really learning what the key appearance and design and what it looks like, because you can't always section exactly the same thing. So the neurons will be slightly different shaped, the direction of the fibers will be slightly different, but the appearance of them will be the same. Just you can't memorize, oh, there's going to be a cell here and a cell here, so I know it has to be the neurons, because they could be anywhere on the slide. So please don't use that as a crutch. Find a way to learn the uniqueness and the shape and not just memorize things like where the cells are located on the picture or the color, are some of the things that are going to fail you if you were just to pick some random slide out from sectioning through each of these organs. So that's the upside of the Quizlet, is I did to be identical to this because I wanted you to actually have the ability to find the key features and not just memorize stuff. But I will be using those pictures on the slide for the Blackboard quiz that I will create and post here, hopefully today or tomorrow, and I'll send out an email to you guys. Is everybody okay with that? This is what it's normally going to be like. It shouldn't be as long as our normal first day lab where we have to get through the syllabus and all the other stuff. But the key is to make sure we get through the key things you need to learn that day, and then also answer any questions. So are there any other concerns? The terminology, bone markings, and this tissue stuff. So for the tissues, you need to know its category. You need to know its name. So like connective, muscular, nervous, or epithelial, then you have to give its specific name. So like simple squamous, areolar, adipose, skeletal. So be careful on the questions. If I ask for the tissue's name and I ask you the tissue's name and the type, that you make sure you include the type would be epithelial, muscular, nervous, connective, and the name would be like simple squamous, areolar. So watch the questions. I may ask you just the tissue generically. I may ask you to include the type or the category it came from. And then, of course, the structures, the word I use for the little stuff would be the structures, are things like the fibrocytes and those smaller parts. And then the last thing I would ask is location. So if I would like you to learn the location that's on that lab handout, if you tweak it and word it slightly different, but again, just remember, I will go back and regrade it. So if you give me kidney instead of kidney tubules, I will go back and regrade that stuff. But try to learn just the ones that are on the sheet, just because it's easiest way to actually quiz you on it and have it be graded correctly. But if you do some minor tweakings to the wording, I will go back and grade it. Just realize your score is going to be lower when it gives you the score originally, and I will just update it when I can get to it as soon as I can. I think that's the main thing. Yeah, just if you haven't tried using your webcam with Respondus, please do so. I fixed the error message on the practice test, so you should be able to use it now. And I mean, it's not a real test, so you can play with it however you want to, just so that you're ready to use that program this coming week up until midnight on Sunday for those Blackboard quizzes. Anything else? Are we okay? I think that's about it. So if you're okay with that, I will let you guys go. I'll send an email out as soon as I have the Blackboard quizzes posted. And then I will get Skin if you want to work ahead, ready to go, so that you have that ready for Monday. Carla, do you have a question?
Yes, I do have a question. So for the Quizlet, I wasn't yesterday I was in the class, but your video had messed up yesterday where you was trying to explain the Quizlet.
Oh, were you at the 1 o'clock one?
Yes, ma'am. Where
Yeah,
your
unfortunately, I lost my power for about 4 or so hours yesterday, and it was, of course, in the middle of that recording, and I was like dead. Power went out. It got nasty here. The wind was going horizontal. Yes, ma'am. So go ahead. Quizlets.
so
Yes.
for the Quizlet, I finished it yesterday, and I was just trying to see exactly how is it transferred over because all it said when I got done was that it was finished.
Yeah, it should
So
say finished, 100% correct, something like that. Congratulations, you I'm all right. Or it gives up some kind of thing. So the biggest thing is to watch anytime that kind of countdown on the left-hand side says you've got all of them correct and 0 incorrect, then you're good.
Okay, so it doesn't matter how many rounds you go through.
No,
Just
it's going to divide it up. Correct. It's going to divide up in rounds just to try to give you a break.
okay.
So the rounds don't matter. At the end, it will generally, I think somebody said it gives you like, you got 86% or you got 75%.
Yes, ma'am.
That's out of the total times you answered the questions. It's
Mhm.
taking all the questions you answered and how many you got right to give you that percent. That percent
Mhm.
has no importance to me. I don't
Okay.
see that. It doesn't tell me any of that information. All it
Okay.
tells me is that you took the time to go through the Quizlet and do them all until you got every question right at least once.
Okay then. Okay. Thank you for that.
No problem.
All right.
Everybody else okay?
So the Quizlet due Thursday for the muscle tissues.
For tissues, yes. So there's 4 types of tissues, so not just muscles, but yes, all the tissues
And
are
then
due. That Quizlet
Sunday,
is due Thursday at midnight, so tomorrow at midnight, correct? I'm sorry?
and then the Blackboard quiz on Sunday.
The Blackboard quizzes are always going to be due Sunday at midnight, correct?
Okay.
And I will send out an email as soon as I get those posted, so you can take them early. As long as you get them done by Sunday at midnight, it's fine. You're free to take them early. Or if you want to leave until last minute, that's your choice. Just make sure you get it done by midnight. So,
All right.
no problem. Okay, so I'll let you guys go. Email if you have any other questions, and I will talk to you, I guess, Monday at 10 o'clock.